Last lecture of the week, at least for me, maybe not for everyone. Hope everything's going well. I have scheduled a review session. It will be on Sunday. Oh. It doesn't matter if I said any time, there'd always be some unhappiness, and I understand that. There's just too many people to get it set up for everybody. Sunday at 1 p.m. in ALS 4001. Okay? Sunday at 1 p.m. in ALS 4001. I will videotape it, so if you can't come, send questions with your friends or whatever, and we'll go with that. Okay. Last time I got a little hurried at the end, so I want to go back and spend a little time making sure that the points that I made at the end get drilled into your heads and that you understand the things there. This was a place where I started getting a little quick, so I wanted to come back and just say a little bit about it. Not too many points to make here, but I did make the point that coenzymes and cofactors, and by the way, I use that term interchangeably, coenzymes and cofactors. I think they're pretty much the same thing as far as I'm concerned. The coenzymes, cofactors, which include, of course, the metals that you can see below, are things that help enzymes to catalyze the reactions that they catalyze. So an enzyme will bind to these things. Sometimes the binding of the coenzyme cofactor is covalent, meaning it's held there and it's not going to come off. In other cases, it's not. It's just simply held in place and it can come on and come off. So it varies a fair amount. We'll see some of these play very important specialized functions. One of them we'll see today, the nicotinamide adenine dinucleotide, otherwise known as NAD, is a very important coenzyme in oxidation reduction reactions. So that's, in fact, if there's something that you want to take away from what I'm saying here, both NAD, that one, and the flavin adenine nucleotide, which is called FAD, both of those are involved in oxidation reduction reactions. And yes, I do think those are important to know. Coenzyme A is what I like to think of as a handle. Cells use this handle, and enzymes use this handle to hold on to things. So the handle provides a way to hold on to fatty acids. So we see acetyl-CoA where it's holding on to an acetate, or we see palmitoyl-CoA where it's holding on to a palmitate. That common handle allows the enzyme to know that what's attached to it is a fatty acid. So that's a very useful thing. Biotin is a cofactor that's involved in reactions where carbon dioxide is put onto another molecule, and those enzymes will always be named as carboxylases. And you see that pyruvate carboxylase is one of those that uses a biotin cofactor. Those are the main ones that we will talk about. Next term, we'll talk about tetrahydrofolate, and we'll also talk a little bit about thiamine pyrophosphate, but those are the main ones that we'll talk about here. At the bottom, you see the metals, and the metals are involved in a variety of reactions, and they're usually known as trace metals, with the exceptions of calcium and magnesium. Most of those metals are needed in very tiny amounts. And some of those metals are actually poisonous. So it's a yin and yang, a little bit good for you, more than a little bit bad for you. Prime example of that's selenium with glutathione peroxidase. Okay? You need selenium for certain proteins that you make. However, anything more than a trace of selenium is poisonous. So I don't recommend, for example, taking selenium supplements unless there's some compelling reason your doctor has told you to do so. Because few people ever are deficient in selenium. Okay. The next thing I talked about was introducing this more complicated topic, and that was the double reciprocal plots, or as they're also known as Lineweaver-Burk plots. Okay. So Lineweaver-Burk plots, as I briefly said last time, are ways of representing data that are different from the way we saw originally. The way we saw originally, we had V versus S, velocity versus substrate concentration. And when we did those plots, we basically had a hyperbolic plot, kind of like the plot that myoglobin showed for binding of oxygen. Okay? Most enzymes will show a hyperbolic plot if you plot them as V versus S. Which ones won't? Anybody remember? Which enzymes won't show a hyperbolic plot for V versus S? Allosteric enzymes, right. What kind of a plot do they show? Sigmoidal, right, okay. All right, so, but most enzymes are going to show a hyperbolic plot for that. If I have a hyperbolic plot, and I take all of the data and I invert it, I take the reciprocal of that data, okay? And then I plot that as the reciprocal data, 1 over the velocity, 1 over V0, versus 1 over the substrate concentration. That hyperbolic plot transforms into a linear plot, as you see here. Now, this linear plot, okay, is very useful. When I was doing the hyperbolic plot, remember that Vmax, when that curve went up, I had to guess where it intersected that line. So I had to guess where that actual Vmax value was. I didn't know where it was. This plot enables me to precisely establish Vmax because it's the reciprocal of the y-intercept, 1 over Vmax. So if I take the value that's shown there at the y-intercept and take the reciprocal of that, I will get Vmax, because 1 over Vmax is Vmax. All right? So the beauty of a straight line is it's very easy to draw. Least squares analysis, I can get that with my calculator computer very easily, and bang, I know the value of Vmax. If I extrapolate that line into the negative regions, okay, then I see that the x-intercept is equal to minus 1 over km. So if I take minus 1 over that value, I will get Km, because minus 1 over Km is Km. So I have 2 parameters of an enzyme that I talked about last time that now I have a very easy way to determine their value. Okay? You might say, well, why don't those points come down into the negative region? And the reason for that is it's kind of hard to have a negative amount of substrate, right? So I have to extrapolate, which means just draw the line all the way down to the x-axis. Make sense? Questions? Everybody's ready for weekend, aren't they? Everybody is ready for weekend. Nobody? Okay. Pop quiz time then, right? Oh, I don't do pop quiz. Okay. So I said double reciprocal plot. You can see the various parameters on there. Linear for enzymes, okay. X-intercept, direct reading of those 2 parameters. All right. The last thing I want to say on this particular lecture had to do with other things that catalyze reactions. Now, other things that catalyze reactions blew our minds when we first discovered them. It blew our minds because the sort of dogma had always been that enzymes are proteins. The only things that catalyze reactions are proteins, right? And then there was a professor named Tom Cech who was working with a lower eukaryotic organism known as tetrahymena. And he was working with a small RNA within tetrahymena. And we'll talk about it next term, but there's a process called splicing where a piece of an RNA is removed and the 2 ends are joined together. And so he was studying this thing, and he was interested in the splicing of this RNA. And he gets looking at this RNA, and he takes it, and his graduate student takes it and hands it to him, and he says, here's the RNA. So when they analyze the RNA, Tom Cech says, you're an idiot. You gave me the spliced RNA. He said, no, I didn't give you the spliced RNA. Yeah, you gave me the spliced RNA, right? So go back and isolate it again. So he goes back, he isolates again, he hands it to Tom Cech, and Tom Cech analyzes it, and he says, you gave me the damn spliced RNA again. I know I didn't do that. Well, then you're somehow contaminating it with the enzymes that make the splicing in this RNA. So this goes on for a while, and no matter what they do, no matter how hard they try to keep proteins and other things away from this thing, he always ends up with the spliced RNA. Now, this is why I want people thinking out of the box. When you think out of the box, you start thinking, well, I wonder if there's another explanation for what's going on here. They scratch their heads, they get to that point, and somebody says, well, maybe it's catalyzing its own removal. That was a pretty radical idea, and it is a pretty crazy thing to think about. It's kind of like doing surgery on yourself and amputating your arm, right? Not something you probably want to do. You might be capable of doing, but you probably don't want to do, right? And so they had the idea that perhaps, in fact, this was splicing itself. So they went and they chemically synthesized this RNA. So there was no exposure to proteins whatsoever. And when they analyzed it, they discovered that it did remove that little intron out of itself. An amazing thing. And they had discovered something that everybody thought, that can't be. That RNA is catalyzing a reaction. It's catalyzing a reaction on itself. Is it crazy then to think that it might catalyze a reaction on something else? Okay? Are there RNAs that can catalyze other reactions? And they started looking, and sure enough, they found other RNAs that could catalyze other reactions, not on themselves. So like when we talk about the catalysis that's involved in the V versus S plots that we see, and we make a product, in each case we were using an enzyme. However, it doesn't always take an enzyme to do it. What they were doing was they were making that with RNAs. The synthesis of every protein in your body, the synthesis of every protein on the face of the earth, this also blew our minds. The formation of the peptide bonds that we talked about earlier in the term, those peptide bonds are formed by a catalytic RNA in the ribosome. Okay? So we had to come up with a new name, and the new name that we came up with, we called these ribozymes. Instead of calling them enzymes, which are proteins that catalyze reactions, we had ribozymes, which were RNAs that catalyzed reactions. Okay? So a ribozyme is an RNA that catalyzes a reaction. We'll talk more about those RNAs that make peptide bonds next term. Okay, questions about that? Hearing none? Okay, that was the metabolic melody from last time. I won't go through that again, but I do have a metabolic melody for today. Instead, right now what I want to do is I want to turn our attention to inhibiting enzymes. Inhibiting enzymes is one of the prime things that people in healthcare are interested in doing. If you want to design a drug to treat a disease and you're trying to kill a bacterium, then if you find an enzyme that the bacterium needs to have alive, and you can find a molecule that inhibits that enzyme and only that enzyme, not all the other enzymes of your cell, you've just created what's known as an antibiotic. I'll give you an example of penicillin later, and I'll talk about that. But antibiotics largely work to inhibit various enzymes. So it's important, if we're going to be talking about things like that, that we need to understand the process of enzyme inhibition. Okay? It's important we understand the process of enzyme inhibition. I'm going to give you two good examples of types, actually three good examples of types of enzyme inhibition. I'm going to leave out the complicated one. You'll thank me for that. I'm not going to talk about uncompetitive enzyme inhibition. You'll see it in the book. I'm not talking about it. You're not responsible for it. And I will encourage you not to read it because it'll confuse you unless I give you some background about it. And I just decided we don't have the time to go through the background for that. Okay? If you want to know more about that, come see me separately. I'll be happy to talk to you. Well, let's talk about the ones that make a lot of sense. The first one is the most intuitive one of all. It's called competitive inhibition. Competitive inhibition involves synthesizing a molecule that looks I shouldn't say exactly, but looks very similar to the normal substrate of the enzyme. I've got an enzyme, it grabs this molecule, it converts this molecule into something else, and I say I want to get rid of, I want to stop this enzyme from doing that, so I make a molecule that looks very much like it. So you can see the competitive inhibitor here is green, and it has a shape very much like the normal substrate. It's not identical, but it fits into that substrate binding site and the active site very nicely. And so what it does is it competes with the substrate for binding on the enzyme. It competes with it. That's critical to understanding this. The competition part of it. All right? So remember, competitive, of course, involves competition. I'm going to show you how that affects the kinetics of this enzyme in just a minute. Here's an example of a competitive inhibitor. We'll talk a lot of things next term. We'll talk next term about an enzyme that converts dihydrofolate into tetrahydrofolate. That was one of those cofactors that you saw earlier. It converts dihydrofolate into tetrahydrofolate, and that conversion is necessary for the synthesis of nucleotides. Okay? There's an enzyme called dihydrofolate reductase that grabs dihydrofolate and makes it into tetrahydrofolate. And if we think about that enzyme, we think cells have to have that enzyme in order to make nucleotides so they can make RNA and so they can make DNA. If you stop that enzyme, you will kill a cell. Do you want to kill your cells? Well, I don't, right? However, cancer cells are also cells, and some cancer cells have the property of dividing more rapidly than normal cells. So if I take an inhibitor, and here's an inhibitor known as methotrexate, and let's look at that structure first. Very similar in structure. It's not identical, but it's very similar in structure to dihydrofolate. Okay? If I treat cells with methotrexate, it will compete with the dihydrofolate for the dihydrofolate reductase. The dihydrofolate reductase will not be able to synthesize tetrahydrofolate, and nucleotide synthesis will be stopped, and I will kill that cell. Now, if it's a normal cell, I'm in bad shape. But if it's a rapidly dividing cancer cell, I've done what I wanted to do. So how do I use this drug? I use this drug for short time periods. In short time periods, I'm more likely to kill a cancer cell than I am to kill a normal cell. And then I withdraw the drug and let the normal cells try to catch up and be okay. One of the reasons people feel very drastic effects from chemotherapy is that a lot of normal cells get affected during the treatment with drugs like this. Okay? Well, the aim is to kill cancer cells, so that's a good thing, right? We want to do that. But we want to make sure that we're not killing normal cells any more than necessary. It's a competitive inhibitor. It resembles the normal substrate. Now, I want to make a point that you're going to lose fairly quickly. So I want you to remember it. The point I want you to remember is that these inhibitors that I'm talking about here, the competitive inhibitors and the non-competitive inhibitors I'll talk about in a minute, these inhibitors do not, underline not, bind to enzymes covalently. Like the other things we've talked about, they bind, they come off. They bind, they come off. Okay? You can imagine a pretty good reason you don't want to bind this covalently, right? Don't you want to give your normal cells a chance to live? So when it comes off and gets replaced by a regular dihydrofolate, then it's got a chance to live after you've killed the cancer cell, right? So these inhibitors are non-covalently bound to an enzyme. They will come off. That means that in the case of a competitive inhibitor, we can outcompete it. And let's see how that happens. Here's a V versus S plot for, on top, number 1, a dihydrofolate reductase with no inhibitor. We see that as we add increasing amounts of substrate, we reach a Vmax. The blue line is the same reaction with a fixed amount. And notice that it's a fixed amount of inhibitor. We don't change the amount of inhibitor because we don't want to have two variables. Remember, we're changing the substrate concentration. We don't want to change the inhibitor. So we have one inhibitor, one fixed amount of inhibitor. What do we see happens with this? We see that at low concentrations of the substrate, that the velocity is somewhat lower. But as we add more and more of the substrate, look how those two gaps are narrowing. What's going on there? Well, the key is that we used a fixed amount of substrate. A fixed amount of inhibitor. As I add more and more substrate, it becomes more and more likely that the thing that's going to bind to the enzyme is the substrate. Let's imagine I get out here to 1,000,000 of substrate to inhibitor. Do you think I'll be able to distinguish those two lines? The answer is no. Okay? The answer is no, because they'll be essentially indistinguishable. And so you can see these two lines are approaching the same limit. And the same limit is what? Vmax. They're both approaching Vmax. So in competitive inhibition, the Vmax is the same as the uninhibited reaction. What happens to Km? Well, you can see it pretty graphically on there. Km goes up, and you said, but Kevin, you said Km is a fixed quantity for an enzyme. How does Km change with an inhibitor? And I told you last time, I gave you the clue. The clue was that the apparent Km is changing. What does the apparent Km mean? Okay. Km is the substrate concentration it takes to get to half Vmax, correct? Okay. At low substrate concentration, what is the relative amount of inhibitor to enzyme? High or low? High. More likely that the enzyme is going to be bound to inhibitor, correct? More likely that the enzyme is going to be inhibited by the inhibitor at low concentrations, correct? At high concentrations of substrate, the ratio of substrate to inhibitor is high, or the ratio of inhibitor to substrate is low, depending on which way you want to look at it, right? At high substrate concentrations, it's less likely that the enzyme will be bound to inhibitor. I'll have a smaller percentage of the enzyme inhibited. Everybody with me? Let's imagine down at the lower level where I have Vmax over 2 that I've knocked out 50% of the enzyme. Is it going to take more substrate concentration for that 50% of the enzyme to reach Vmax over 2? It is. For that 50% of the enzyme, each one of those, do you suppose its binding affinity is affected by the inhibitor? It's a good exam question, guys. I'm asking you now about, I've got this mixture, got the enzyme, I've got inhibitor, I've got substrate. 50% of the enzyme is bound to inhibitor. 50% of the enzyme has no inhibitor. I'm asking you about the enzyme that has no inhibitor, and I'm asking you if you think its affinity for substrate is different in any way. No, right? However, what's confusing me, this is why it's the apparent thing, is I've only got 50% of the enzyme to give me enough to get there. So I got to really load the substrate up to get to that half Vmax. That's why when we look at this, that the Km apparent increases, because I've got less enzyme to get to that velocity that I need. So, in competitive inhibition, the Vmax remains the same, and lazy people like me say the Km increases, but really it's the apparent Km that increases. Right? Understand that? Questions about that? Yeah. On an exam, I will generally be flexible. However, if you try to tell me that Km is not a constant for an enzyme, then that might cause trouble. Okay. But in general, yeah, I'm pretty flexible with that. Okay. Because I'm lazy about it myself. I say, hey, the Km goes up, right? Okay. What does a Lineweaver-Burk plot look like? Well, it looks like this. Oh my God, we're going to have to do that too, right? Yeah, it's really easy. It's not complicated. You already know the information to draw this plot. What did I tell you? I said Vmax does not change, right? Does 1 over Vmax, is it going to change? Of course not. So you know 1 point of the inhibited line. I told you that the Km, or the apparent Km, was going to increase, correct? What's going to happen to minus 1 over Km? It'll get closer to 0. Now you know 2 points. Do 2 points define a line? Yep. Okay? In the enzyme song last time where it talked about inverting everything and having 2 points to define a line, those are the 2 points I'm talking about. All right? You can memorize this, but you could also just learn it. If you learn it, you don't have to memorize it. And I like to memorize as few things as possible, especially at my advanced age, guys. Okay? All right. 1 over Vmax unchanged, minus 1 over Km increases, meaning I'm sorry, minus 1 over Km increases closer to 0 means that Km itself increases. All right, let's talk about another type of inhibition. This one's not quite as intuitive, but it's not overly complicated. This is called non-competitive inhibition. And non-competitive inhibition, the inhibitors do not resemble the substrate. They do not resemble the substrate. Okay? If they don't resemble the substrate, how do they bind to the substrate binding site? The answer is they don't. They bind to a different site on the enzyme. And you've already seen numerous examples in this class about how binding of something can affect an enzyme's shape and therefore its enzyme's function, right? We've seen very small changes in structure can have enormous changes in terms of structure and function. So it's not surprising then that we might have factors that we could bind to an enzyme that might change the enzyme in a way such that it would change the enzyme's catalysis, and that, one of those factors, is a non-competitive inhibitor. You can see it illustrated on the screen. Okay? A simple depiction of how that works. All right? Now, the very different thing about this type of inhibition is you can't outcompete it. In the first one, we outcompeted it, right? How did we outcompete it? We started adding tons and tons and tons of substrate. And the more substrate we added, the less of an effect the inhibitor had. That's why the two got up to the same Vmax. However, that's not the case here. If I add a ton of substrate to this, the substrate is not competing for the binding with the non-competitive inhibitor. What do you suppose is going to happen? Well, this makes us think about the enzyme in a very different way. Okay? This means that no matter how much substrate I add, I will always have a fixed percentage of enzyme inactivated. This is the key to understanding non-competitive inhibition. A fixed percentage. You say, well, it comes on and comes off. Well, as soon as it comes off of one, it goes on to another. So effectively, the same percentage of enzyme is constantly inhibited. Let's imagine that I added enough non-competitive inhibitor to knock out 50% of the enzyme. There's my magic number today. 50% of the enzyme. Okay? Anybody envision that? 50% of the enzyme. Let's say I measure the Vmax for this reaction. Is it going to have the same Vmax as a reaction that has no inhibitor? How many people say yes? No brave souls, they've looked ahead. How many people say no? Okay. Everybody gets an A. Okay. Why is that the case? The reason that's the case is, let's imagine I took, and you can see it right here, let's imagine I took, instead of adding inhibitor to the blue one, let's say I started with 50% of the amount of enzyme. If I started with 50% of the amount of enzyme, wouldn't I get 50% of the Vmax? Because remember, the Vmax is dependent upon how much enzyme I add. So, non-competitive inhibition will have a reduced Vmax compared to an uninhibited reaction. The thing you'll have a little hard time getting your head around is that the Km is not going to change. The Km will not change. Now, why did it change before and it's not changing here? Or why did it apparently change before? Good professor, good professor. Okay? Any ideas? I actually told you the answer earlier. Yeah. No, that relates more to the Vmax. So he said, you know, because more substrate doesn't have any effect. But that actually relates to the Vmax, not to the Km. Yes? Inhibited enzymes can't bind substrate at all. I can only measure the Km for those that bind substrate. Well, what's different from before? What's different from before was we had varying amounts of inhibition. That's what I told you before. Here we have a constant amount of inhibition. We're not confused by the amount of enzyme being active, which is what confused us before. Here we have a constant amount of enzyme, and I can only measure Km for active enzyme. It got thrown off last time because the amount kept varying as I added more and more substrate. It doesn't vary here. So Km is a constant for an enzyme, and I can only measure Km for active enzyme. So just like before, if I try to measure the Km of the enzyme without an inhibitor, will its Km be the same? The answer is yes. But now the difference is I have a fixed amount of enzyme, not a variable amount. Okay, well, if we imagine now this in terms of Lineweaver-Burk, okay, what's going to happen? It'll be there in a second. All right, Km is not affected. Here's what it looks like. First of all, we said the Km was not affected. So that means minus 1 over Km is going to be the same. There's one of our points. And notice that the upper line is the inhibited line here. Huh? Why is the upper line the inhibited line here, guys? Why is the 1 over Vmax higher? It is the reciprocal, of course. So a smaller Vmax means 1 over the reciprocal is going to be a higher number, right? Bang. I now know what that line is going to look like for the inhibited compared to the uninhibited enzyme. Okay, a lot of stuff there. Questions about that? Clear as mud? Yeah. In terms of drugs, are there any advantages a competitive inhibitor might have over a non-competitive inhibitor? It's a little hard to say. There's a lot of variables that goes into these, and what happens with drugs isn't so much the individual enzyme that you're trying to get, it's the effects on other things that cause drug side effects. That's where the problems arise with drugs. So the main thing is, first of all, finding something that inhibits an enzyme, and then second, seeing if it has effects on anything else that's in the cell. So there's nothing inherently better about one versus the other. We'll see later that when I talk about allosterism, that allosterism follows a model very much like non-competitive inhibition. But in terms of advantage for one versus the other, the answer is really no. Okay. There they are. There they are. All right. Here's a cool one. This is a different kind of inhibition. This is fundamentally a different kind of inhibition. It's called suicide inhibition because it is a covalent type of inhibition. Covalent. The inhibitor makes a covalent bond with the enzyme and kills the enzyme because, as you know, covalent bonds are not easily broken, and once that happens, the enzyme is going to be stuck with the inhibitor. The best example of that is penicillin. Penicillin, of course, we know is an antibiotic. And by the way, antibiotics, as I briefly said earlier, are things that we use that specifically target something of a target cell or a target virus, okay, without affecting the enzymes of a regular cell. Well, bacterial cells have a cell wall. We don't have a cell wall. Okay? Our cells don't have a cell wall. Bacterial cells have to build a cell wall, so if we target something that makes the bacterial cell wall, we're much more likely to find something to kill bacteria but not affect our cells. So that's what happens with penicillin. Penicillin actually binds to an enzyme called DD transpeptidase. No, you don't need to know that. Okay? DD transpeptidase is responsible for making the cell wall of bacteria. And penicillin looks like the normal substrate of the enzyme. So it's kind of like a competitive inhibitor, but the difference is that instead of competitively inhibiting, which would mean it could come off, it makes a covalent bond with the active site, and the enzyme can't shake it. Well, if the cell can't make a cell wall, the bacterial cell is basically unable to function, and the bacterium dies. Okay? So suicide inhibition has some advantages as well. And in the case of penicillin, that's how it works. Okay. Now, I want to introduce a term I've mentioned a couple of times, and this is a very important concept. We'll talk about it, especially beginning on Monday, about this. The concept is called Allosterism, We'll talk about allosteric effectors. We've already mentioned it in a song. Okay? Allosteric effectors. So what is allosterism? Allosterism is a phenomenon where a small molecule binds to an enzyme and affects its activity. A small molecule binds to an enzyme and affects its activity. Some people say that it binds to a protein and affects its activity, and I'll accept that also. So I don't worry about the dividing line of that. Okay? Binding an enzyme and affecting its activity, binding a protein and affecting its activity. Same thing as far as I'm concerned, okay? Now, that turns out to be a very, very effective tool, as we'll see on Monday, for cells to control metabolic pathways. I've been describing enzymes as things that are race cars. And race cars are great for speed, but they're not so great for safety, as I mentioned. Enzymes are race cars. They're great for speed, but they're not really great for safety. So we need to make sure we carefully control those, and allosterism is one of the ways in which we control enzymes. Okay? Notice it's the binding of a small molecule. By small molecule, I'm talking about something like a substrate, or something about the size of a substrate, not a protein. There are two types of allosteric effectors that we'll talk about. One is called a homotropic effector, and homotropic effectors are substrates for the enzyme. A homotropic effector is doing two things. One, it's binding to the substrate binding site on the enzyme, and two, it's affecting the enzyme's shape. And you know by affecting shape, it affects activity. Now, I should emphasize to you, when I talk about allosteric effectors, or I talk about allosterism, these can operate in a positive sense and in a negative sense. In a positive sense, we describe them as, quote, activating an enzyme. In a negative sense, we describe them as, quote, inactivating an enzyme. And I put those in quotes because it's more like we're turning the volume up and turning the volume down than absolutely turning them on. It's not a light switch. We're not turning it on or off. It's more like a dimmer switch, right? My dimmer switch is on pretty strong right now. I'm kind of dim, but all right. Dimmer switch. That's more like what allosterism works at. We'll talk about other mechanisms to control enzymes that are on-off switches, but allosterism is mostly an up or down, not an on or off, although we do use the terms activate and inactivate. All right, the other type of allosteric effector is a heterotropic effector, and that's the most common type. It's a non-substrate. Okay? A non-substrate. And I won't give you examples of those today. We're going to see several examples of those on Monday, and so I'll lead you through those and remind you what those terms mean at that time. Okay. Models of allosterism. So let's briefly go through the models. You've actually seen a little bit of this already. The sequential model. You saw that. Where did you see this before? Hemoglobin, right? The binding of oxygen. And I told you that the binding of oxygen by hemoglobin was very much like the binding of a substrate by an enzyme. Surprise, surprise, the sequential model is the same for the two. The sequential model says that there's a cause and effect between the binding of the substrate / effector and the enzyme. And that this binding causes a T to R or R to T, it can go either way, change. Okay? The important thing here is the cause-effect. Cause-effect. The binding of the substrate or effector, whatever we want to call it, causes a change, RT. Okay? I'm going to show you two other models where there's not a cause and effect. All right? You've seen this before with hemoglobin. There we are in the T state with the squares. We can see this guy changing, and we can imagine that that L on the very second one from the left has bound its first substrate. And we can see that the binding of that first substrate converted it into an R, and we see the two things around it starting to convert into R state. And when they start converting, they become more likely, in the case of hemoglobin, to bind more. So we can imagine this being, in this case, an activation mode, right? But there's a cause and effect. The binding of the first one caused the second ones to start changing shape. Okay? That's the sequential model. Okay. Another model called the concerted model, also called MWC, which are the initials of the people who proposed this model, looks like this. We see the enzyme existing in two states, and again, these are all multi-subunit enzymes. We see the enzyme existing in two states, a T state and an R state. Okay, that's just like before. Well, not quite. Before, the substrate or the effector caused the change. In this case, the change happens independent of the substrate or the effector. The enzyme is flipping all on its own. In solution, this model says that enzymes flip completely between the T state and the R state, and that what we see as allosterism arises when a substrate or an effector binds and locks it in that state. So instead of being caused, the state is being locked by the substrate or the effector. Okay? This is the MWC model. It's a little bit conceptually more difficult to understand. Well, how does the enzyme flip? Don't worry about that. Okay? And by the way, a model is simply that. It's a way of explaining something. Models aren't necessarily the answer. Models explain data that we see. They explain phenomena that we see. Right? But they're simply that, an explanation. Explanations, you know, aren't always perfect. Okay? But it's a way to explain how the T and the R states arise. That's what this model does. Okay. The last model I want to show you is called the morphian model. The morphian model is related to the concerted model. The difference being that the morphian model says that the flipping that happens not as one big complex, but first the complex comes apart, then individual units flip, and only after the individual units have flipped can they bind to each other. Okay? So the concerted model says the whole thing flips on its own. This thing says it happens in pieces. But also, like the concerted model, this says that the effector locks it into that state once it's there. So it's not a cause, it's not a cause and effect. The difference between the concerted model is it's simply happening in pieces instead of happening all at once. And there's a lot of enzymes for which this model appears to be correct. Okay, questions on the models? Okay, yeah. Yeah. At what stage does it land and lock it in there? It's a very good question. It actually could land and lock it in either of the single modes, or it could actually lock it in the complex mode as well. So there's not a specific place where that locking might occur. But we can imagine that that locking would make it act like the concerted model, and this does act something like the concerted model. This simply explains it in smaller terms. Other questions? Good question. Over here. Yeah. So the question is, do we know which model explains it the best? Some enzymes appear to be consistent with one model. Other enzymes appear to be consistent with another model, so there's no one model. And some enzymes appear to actually be a blend of models. So that's probably not the answer you wanted to hear. He said, Kevin, give me the most important one, and I'm going to go with that one, right? But it turns out that the reason we have different models is that different enzymes exhibit different data behaviors, and there's no one model that explains all this. But there are enzymes that show examples of all three of these different models. Suffice that to say. Okay? Yeah? So in the middle of this figure, the question was, what do I mean by individual units? In the middle of this figure, you see an individual die, okay? And so those are the individual units, whereas the complexes are on the left and right sides. Okay? Okay. Well, question? Oh, yeah, yeah. So your question is, are these more with heterotropic versus homotropic? In general, the heterotropic models heterotropic effectors are more common. But I won't say any of them are unique to any of these particular models, no. Yeah? Yes? So when they're down to their individual subunits, will they be, that's a very good question actually, will they be able to carry out their normal activities? And again, it's going to depend on the enzyme. Most likely, most enzymes are going to be able to do that, but it's not an absolute thing. It's not an absolute thing. So we're using this model as a way of explaining activity, up and down activity, and that's really more at which the models come into play. Okay? But very good question. Okay, let's see. I'll tell you what. People wanted to know where we're going to stop for the exam. And though I've got a little bit of stuff to talk about here, I think I'm going to save that for the next exam. And I've got a song for you that I want to go through. I'll come back and talk about this stuff next time. This is another one of those songs that it's really difficult for me to sing, so I've got a professional singer who has recorded it. I'll try to add to it. It's a beautiful song. It's called Catalyze, and I hope I can get your attention for it. To the tune of an old Carpenter's song. My enzymes truly are inclined to convert things they find. Turn the key covalently. Catalyze. How do cells regulate these roles? Allosteric controls. Two forms C, states R and T, modulate. Inhibition keeps the substrates from the active site. They raise Km but leave Vmax and sure. While the non-competititves find us where and lift the plot fade on Lineweaver-Burk. Other ways enzymes can be blocked when things find and get hot. Not free, tied to the key, suicide. Penicillin's action stops peptide-like and cross-links in bacterial cell walls in awesome ways. Beta-lactam rings react as such, starts bonding with the transpeptidase. So there are several enzyme states counteracting substrates. Now you see, blocking the key regulates. This have to be controlled. Some get slow, put on hold. It's sublime how the enzymes catalyze. It's sublime how the enzymes All right, guys. See you.